lab instructions, I'm not emailing them this week. I put them on my Wartburg under the syllabus section. I don't know why I didn't do that before. I do it for everything else. Okay. We'll go ahead and get started then. Today we're talking about genetic inheritance, patterns of genetic inheritance. And this is pretty fun stuff for me. I kind of mentioned that at the end. I like to do genetics. So I did my master's thesis on genetics, did my undergraduate research on genetics. So this is like my favorite stuff to do. So I'm a little bit biased. I find this stuff very interesting. So we're going to go ahead and get started. It's chapter 21 in your book. So the reason we want to know about patterns of inheritance, there's some simple things and then there's some complex things. So it's kind of interesting to know, like, your blood type. If you knew both of your parents' blood types, you could find out the possibility of the parents and the probability of what their children's blood type would be. You could figure out whether or not they'd have a certain hair color or hair type. And by hair type, I mean like coarse or curly or straight or things like that. Thin hair, thick hair. All these different things about us are things that were genetically inherited from our biological parents. Again, we start with those points to ponder. A couple of those words that we're going to be talking about today are genotype, phenotype. Maybe you've heard of those words before. We're going to talk about different ways that genetics, the patterns of inheritance. So there's things called sex-linked inheritance. There's things called X-linked inheritance. The difference between dominant and recessive. We're going to do some Punnett squares, which if you like puzzles or Sudoku puzzles, they're easier than Sudoku puzzles, but they are ways that we figure out genetic inheritance. So a couple of questions, and maybe if people are on Zoom, they could add this to the chat. The first question, and it can be a yes or no question, is do you have a widow's peak or a straight hairline? Maybe you're going to have to pull out your phone and take a selfie. You probably know if you have a widow's peak. So a widow's peak is kind of like where your hairline comes to a point. And sometimes if you have like a shorter haircut and it comes down here, maybe you have to kind of pull back. I do not have a widow's peak. Let's see, does anybody in the chat for Zoom, does anybody have a widow's peak? Doesn't look like we got any widow's peaks on there. So widow's peak is an inherited trait. Okay, the next one is, are your earlobes attached or unattached? So I know it's probably hard to see mine because I'm wearing a mask, but mine are not attached. So mine has like a loop. Here's a talk. So my ear kind of looks like that, and then my face looks like that, and then I have a little smile, and that looks like that too. Okay, if they were not attached, it kind of just goes right into your face, where they're not. And so some people are different in that sense. Doesn't give us any advantage or disadvantage. It's just how we inherited that. Does anybody have an attached or unattached earlobe? I see it. Maybe. Lauren, let me see that ear. I think you've got an attached one. Oh no, it's unattached. All right. The next one is, do you have short or long fingers? Okay, so shorter fingers. I would consider myself to have longer fingers, even though in relation to other people, maybe they're short. If anybody knows my sister, Annie, you can make fun that she has short fingers. Do you have freckles? Freckles is inherited trait. Anybody have freckles on the call? Anybody got freckles here? Yeah, awesome. And we'll also see with some multi-inherited traits that some traits are kind of linked to each other. So most of the time, but not always, freckles come with, maybe you have a little bit finer colored skin, or maybe you have lighter colored hair. Not always, but that's just some of those things that are possibly linked together. All right, so the word genotype. The word genotype and phenotype are going to be used to compare different things. So genotype and phenotype are spelled different ways. Genotype are the specific genes for a particular trait with written symbols. So the way, sorry, I need to go back to sharing my screen for the Zoom folk. Okay. All right. So the genotype, if let's say the trait for, let's say brown hair, just for the example, this isn't maybe necessarily the right thing. If you have brown hair, okay, brown hair, let's say that the genotype for that, it's dominant to have darker hair. So we're going to say the letters are big D, little d, or big D, big D. I'm going to write that on the board for the people here in the classroom. So let's say that this, if you have brown hair, let's just for this example, say that it's a dominant trait. So if something is dominant, it has to have those big letters. Maybe you've done this before in high school. So to have that trait, you've got to have these. Now this is a dominant and this is a recessive. These are two dominants, okay? The genotype is this. This is a genotype. The letters, the symbols, these are the genotype. I'm going to highlight that for our folks back home. That's the genotype. Okay, so those are those specific written symbols. The word phenotype is describing what the physical trait is. So when I see the word genotype, I think of gene code, G-E-N-E space C-O-D-E, gene code. The alleles are the alternate forms of a specific gene at the same position, the locus on a gene. An allele for an attached or unattached earlobe, the alleles occur in pairs. So this is one allele. They come in pairs. So this is a dominant allele, dominant allele, dominant allele, recessive allele. So the individual letter here is considered, oh man, I was trying to draw here. Let's see. Didn't work. Let's do pen. This is a specific allele. This is a dominant allele, and this is a recessive allele. So the word is allele. So alleles come in pairs. Occur in pairs. There we go. Just catching everybody up here. So we have a dominant gene and a recessive gene. We indicate a dominant gene by a capital letter. So here's some examples. Big T, little t, and big T, big T. These are both dominant. Big T, little t, even though it has two letters, think that the dominant, the big capital letter, always overpowers the lowercase letter. Okay? When somebody has two different, so big T, little t, they are considered heterozygous. The root word being hetero. And we're going to talk about that in the next slide. When you have something that is the same, like big T, or little t, those are considered homozygous. So you can be homozygous dominant or homozygous recessive. The one that you guys can't see on the board in class here is recessive allele. It is only expressed when the gene has two of the same type. So in order to be recessive, to show the recessive trait, you have to have two small letters. When we work through these examples in class and on your test, it's really important that you use letters that the capital and the lowercase letter look different, okay? So using the letter D, T, B, A, those are all great examples. Try not to use S, right? Because you're going through it with most of our exams are going to be online, but like when you're working through something, don't use the letter S, because if you start to kind of like get a little lazy and it kind of looks like that, how am I supposed to know that it's two different letters? Don't use the letter O, even though that kind of looks, one is bigger than the other. It's hard to tell, especially if you're trying to show me if something is recessive or dominant and it was homozygous. I couldn't tell if that was recessive or dominant. So some good examples would be use the letter T, use the letter A, B, D, I feel like an elementary school teacher. All right, so you've got some examples there. You can use the letter R. Things that don't look the same. It's going to be easier for you in the long run. I know it's sometimes fun to like use the first letter of your name and stuff like that. Mine happens to work out, but just make sure you're using a letter that the two look different. Okay, so these are those three words I talked about previously, homozygous and heterozygous. When in doubt, look at the root word. So the root word being homo and hetero. Homo meaning same and hetero meaning mixed or different, okay? If you are homozygous dominant, you should know that that person has the same letters and they're dominant. So it's got to be two big T's or two big A's like it shows there. If you are homozygous recessive, well, if you're recessive anything, it has to be homozygous. You can't have a big T and a little t and show that recessive trait. It's got to be little t, or little a, little a. Homo meaning same, recessive meaning the small letters. Heterozygous, you always show the dominant allele. So you have the capital letter here. Heterozygous means different. If you are heterozygous, you show the dominant trait, but you are a carrier of the recessive trait. Remember kind of last time we talked about sickle cell anemia and being a carrier? Being a carrier for a trait doesn't mean that you show it. It just means that you're kind of carrying it along with you, and it's possible that you could pass it down to your offspring. That's why if you have parents who, for this example, maybe both of them are brunette or have brown dark hair, and one of your siblings has blonde hair. Possible, right? Not every situation is like that, but in that example, it probably means that somebody was carrying that recessive trait even though they weren't showing it. Okay? Now, the opposite word to genotype is phenotype, and the way that I remember the word phenotype is it starts with a PH like the word physical. So this is the physical traits. Remember gene code and physical traits. So the genotype is like it shows on the left-hand side there. Those are the letters. Those are the alleles. These are the code that tells us what the phenotype is. Okay? So whether or not you have an attached or unattached earlobe is in wording the phenotype, and the code for that is the genotype. Like it says, the physical or outward expression of the genotype. So it says, what are your genotype and phenotype? Well, if you have an attached earlobe, which I don't think anybody did unless they're just not wanting to share, and that's cool too, you're for sure a recessive. If you have an unattached earlobe like mine, then you are either a dominant homozygous or heterozygous. There's no way that I could tell that unless I got some sort of genetic testing, and honestly that's pretty expensive to figure out whether or not my earlobe is attached or not, but you know, to each their own. Okay, so now we have a little bit more understanding of genotype and phenotype. So here's a picture of a sperm and an egg cell, and you have a trait here for albinoism, and this is a lack of pigmentation. You've maybe seen some pictures of animals before that are albino. I'm on page 454 if you're trying to follow along in the book. So it says that generic inheritance affects our characteristics. So whether or not you're normal or you don't have albinoism, it's a dominant trait. So if you are albino or you have albinoism, you have recessive alleles. The way I can tell that is right here at the bottom of the picture. You guys can kind of see that here. It's so hard to draw a circle on this. Okay. So you have a sperm and an egg cell. We are normally called diploid, and you guys don't need to know this word it might mention it here at the end of the slides. I don't remember. Diploid, it means that we have two pairs of everything. Those are our alleles. That is two, so hard to draw on this, and okay. Sperm and egg cells are called haploid. And it's important that sperm and egg cells are haploid because they have half, or they have one allele on each of them. If they didn't, you'd have a sperm cell and an egg cell that both had full copies, and they'd come together and they'd make 4, right? 2 plus 2 is 4. A haploid, if I can write this up here, haploid, I guess I could just write it on the board too. Yeah, we're gonna write it on the board. Let's ignore that crazy handwriting. Okay, so we have diploid, which equals 2n, and haploid, which equals 1n. And this is like sperm and egg cells. And this is like our regular cells. So like our skin cells, our tissue, whatever, those are regular, okay? So here you can see on the picture that the sperm has a couple of options here. It can carry a big A or it can carry a small A. I'm gonna write this here for you. Okay, the egg can either carry a big A or a small A. We can see that at the top. And so when they come together, they have the opportunity to have these different types of scenarios. If the sperm and the egg are both big A's, you show the dominant trait, and in this case, you're normal. If the sperm and the egg come together and they're both carrying a recessive trait, you show that recessive trait, and it ends up being albinoism. For this example, it can be many different traits. Then we have big A, small a, where the sperm here is carrying a small a, but the dominant egg allele is still going to show that they are normal. So that's just an example. Here we have short fingers versus long fingers, freckles versus no freckles. These are just other common inheritance things that we talked about in that first slide. Just some more examples of different traits. Some of these traits not giving us any advantage or anything like that. They're just how we are built. So now we have crosses. Now we're talking about when we have a sperm and an egg cell, or two individuals who at a certain location on their DNA carry a certain trait. When we're looking at the inheritance of one characteristic, let's just say it's short fingers, okay, or long fingers. The opposite of that is a two-trait cross. And that's kind of what I was talking about a little bit, where the traits are kind of carried along with each other. It's hard to think of a lot of examples because nobody really fits into a perfect box. But when you cross two different traits that are related to each other, that's called a two-trait cross. So it says gametes only carry one allele. So the gametes are what is called, for the sperm and egg cell, that's another way to say gamete. And they show what's possible, but the gametes carry that genetic material. And when a sperm and an egg cell are fertilized, that genetic material is crossed. Okay, it says, so if an individual has the genotype big W, little w, what are the possible gametes that the individual can pass on? So let's think of it this way. Let's think of it as an egg cell. Just for the example, you can also say it's a sperm cell. And here, I didn't make this PowerPoint, this is from the book, but I would not have used big W, little w, because both W's look the same. Okay, so we've got, let's see here, big W and little w. The alleles that are possible to pass on are both. They could pass on a big W, or they could pass on a little w. So you as an individual at this specific location for this trait, you have the dominant trait, whatever this trait is. Your gametes, or this individual, doesn't have to be you, your gametes have the possibility of passing down a small w, even though you don't show that trait because you carry it. So the answer is big W or little w, but not both. So here's an example of freckles and non-freckles. Here, both of the parents, so the parents is that top left box here, and it says no freckles and no freckles. So both parents do not have freckles. This is a recessive trait. That means that all of the sperm and all of the egg cells are all going to look the same. There is no possible way, so that's some sperm, here's some egg cells. I'm doing some really artsy stuff here on the Zoom. Okay, that means that there's no possible way that their offspring could then have freckles because all of their gametes are small f. The way we can kind of figure this out is by doing a Punnett square. And I'm going to show you an example of these Punnett squares on the next couple of slides. I recommend that you try and do it on a scratch piece of paper near you while we work through it, because it's something that's easier to just do than it is to explain it. So it's a 2-grid system. So what you do is you put the possibilities on the ends of the square. Some people like to keep it consistent and always keep the female on one side and the male on the other side. It doesn't matter as long as you do it right. All that matters is that you worry about the intersections, okay? I've also said that this doesn't mean that your first child is going to be big F, and then your second child is going to be big F, small f. This means that every time there is offspring, this is the possibility. There is a 75% chance, you guys can't see it, but the dark colors mean that it has freckles. There's a 75% chance that their kids will have freckles. Okay, where did I get that 75%? That 3-to-1 ratio. Okay, so 3 out of 4 or 3-to-1 are going to have freckles. So let's talk about that. So the female on the top is big F, small f for this trait, meaning that the female has freckles. Her genotype is big F, small f. The male, or the sperm cells, have the possibility of being big F, small f. Okay, because that individual has freckles, but they are heterozygous at that trait. So these are individual alleles. They're also individual eggs and sperm, if you think about it. And I'll draw that on this, if that helps. So these are individual sperm and eggs. And the probability of that is what we're going to find out here, okay? So then you look at the intersections of each square. So you go, okay, the first top part and the first side part, we put them together and it's big F, big F. So that's why in this first top left square, it's big F, big F. Then we do it again for the next one. You have big F and small f. And you have big F and small f. And finally, in that bottom right-hand square, you have small f because of the intersection of the square. Does anybody have any questions on how that is found? Yes.
So you would only know what your F is if I'm too big F, or if I'm big F, little f, by doing a genetic test.
Yes.
You can't just say that you have this just by looking at that. Right?
Correct. Correct. So when you're dominant, if you have the dominant trait, you don't know what your second allele is unless you were to do a genetic test. But you show the dominant trait. And then if you show the recessive trait, you know both of your alleles. It's a good question. Anybody else having a question on Zoom on how we got that? Okay. So last part is that phenotypic ratio, making sure that it's in the lowest possible figures. So if this was a different Punnett square and it was a 2-to-2 ratio, you guys would know to bring that down to 1-to-1 ratio. So that's simplest form, smallest figures. The phenotypic ratio is important because this is the probability of what could happen. Okay, it's possible that these two individuals could have 4 offspring that have freckles. It's possible. The probability is higher that, or excuse me, they could have 4 individuals that do not have freckles, even though the higher probability is freckles. These are just every single time that there's an offspring that this is the percentage. And this happens for every single trait. Okay, so let's practice a Punnett square here. It says, what would a Punnett square involving a male and a female with a genotype of that and that look like? So we have, oh, it's the same one we just did. So same thing here that we just did in the last one. So we'll move on from that. Fine by me. Okay, practicing the ratios, like I said, those phenotypic ratios, the genotypic ratio. This is the number of offspring with the same genotype. Remember, there's 2 different words here. There's a genotypic ratio and a phenotypic ratio. Here, genotypic and phenotypic ratio. So the genotypic ratio is going to look different than the phenotypic ratio because we have 3 options for genotypic ratio and we have 2 options for, wow, I am so bad at drawing on this. I am so sorry. I'm just gonna draw on the board for you guys. So when you have genotypic ratio, you have 3 options. Okay, remember gene code. For phenotypic ratio, you're either trait or trait. No trait or trait. Okay. So in that last example, we had a Punnett square. We had big F, little f, big F, little f. We look at the intersections of each place. Okay. The genotypic ratio, how many had big A, or big F, big F? If that's confusing, I can use it. How many are big F, big F? 1. How many are big F, small f? 2. And how many are small f, small f? 1. This should add up to 4. And it does. Okay. Now, how many show the trait if the trait is dominant? 3. And how many don't show the trait? 1. So the 3-to-1 ratio stands, but do you see how the genotypic and the phenotypic ratio are different? So let me do that here for the Zoom. We had a Punnett square, and you had a big A or a small a, or you can do F's. The letter doesn't matter as long as the concept is there. You had this Punnett square that we had on the last slide. Everybody follow me so far? I'm not going to fill it out, but this is our options here. How many are big F on Zoom? Can somebody answer me on Zoom? How many are big F, big F?
1.
Yes, 1. How many are big F, small f?
2.
Good job. And how many are small f, small f?
1.
Good job, you have 1. So this is the genotypic ratio shown on the next slide. So that's exactly what we just did. You see the genotypic ratio is 1 to 1. and the phenotypic ratio is 3:1. All right, the monohybrid cross. Remember the word mono meaning one is an experimental cross in which the parents are identically heterozygous at one gene pair. Okay, so this is a monohybrid cross where we have the letter A, where the offspring of a monohybrid cross indicates if an individual with a dominant phenotype is homozygous or heterozygous. What you need to worry about here is just making sure you know what the parents are and knowing what the offspring are. So here we have parents that are big F, and small f, small f. In the table, all of their offspring are going to have the dominant trait, and they're all going to be heterozygous. This is the example on the next one where we have small f parent here, which they're here, if you can kind of see that. And then you cross it with big F, small f, and then there's a 50% chance of them being small f, and 50% of chance of being big F, small f. So what you guys are missing down here is 2 small f squares. You can look at the phenotypic ratio there on the bottom, all freckles on the left, and a 1-to-1 ratio on the right. Okay, so this is figure 21.5 in your book. So meiosis results in genetic diversity of gametes. So you've maybe heard of the words mitosis and meiosis. Mitosis is the splitting and dividing and replicating of all of your normal cells. I just got to add somebody into the Zoom really quick. Okay, good. So the difference between mitosis and mitosis, I think of it as like my toe, like literally my toe on my foot. The cells, like your skin cells, everything in your body except your sex cells. Meiosis is the dividing and replicating of your sex cells, your gametes. Meiosis results in genetic diversity because of things called like crossing over and things like that. I'm trying to think of the other term, which maybe you guys don't need to know, and that's okay. We might need to know that, but we'll talk about it in a little bit. So meiosis also has 2 divisions. It's important that meiosis has 2 divisions because we want our gametes to be 1N or haploid. We want them to not have a full number of traits or chromosomes. This is because we want to create offspring that look or have similar traits to us. So 2N versus 1N, we want to have our gametes be 1 copy and not both. So as you see at the top, a cell has 2 pairs of homologous chromosomes. So you can see 2 red and 2 blue. What you can also notice at the top is their different sizes. This is just indicating that it's like a dominant and a recessive allele, okay? In meiosis 1, they come to the middle, they separate and divide. There's the first division, that's meiosis 1. Now we have the same amount of cells. Well, they replicated here, which I should mention. They replicated here, and then they divided. Then they divide again because we want to have the end result, which is what is seen at the bottom of the PowerPoint here, that show that they have 1 chromosome per trait. They have 1 of those alleles. Okay, you guys can't really see that, but it's page 458 in your book on the bottom. Okay, so we have a dihybrid cross, which is the opposite of monohybrid cross. This is a type of 2-trait cross. It's an experimental cross, usually involving parents who are homozygous, we know what that word means, for different alleles of 2 genes. So it results in a 9-3 to 3-1 genotypic ratio for the offspring. Well, that's kind of hard to imagine. Well, the first thing we know what happens is we have big F and small f here as options. You guys can kind of see that over here. For one of the traits, it's big F. For the other trait, it's S. So there are 2 letters here. Our ending genotype will have 4 letters to it. The gametes have a dominant or recessive in this case. And then we kind of just make a bigger Punnett square. Okay? We look at every possibility. So if the individual, and I'm going to zoom in here in class so you guys can kind of see this a little bit better. So this was the page from the other book, okay? This person genotype on the top left is, let's see if I can, that's not what I want. Don't. Push around button. Okay. So we have big F, and small s at the top. That's that person's genotype. Then you have this person over here who's small f, and small s. Their gametes could look anything like shown in this picture right here. So the gametes are on the outside. All the sperm and egg cells, they're on the outside of that Punnett square. See that a little bit better? And then every single cross of that intersection are the possibilities. This is a dihybrid cross. This is kind of a little bit more difficult to understand. So it's kind of thinking like maybe freckles are tied to light toned hair. Not always the case, as we know, but this is a dihybrid cross, meaning that at each intersection, they have a possibility of an F and a possibility of an S. And what are those? And because we added more gametes, the probability decreases of each trait.
So basically, you have to do 2 Punnett squares or a Punnett square before you can do this one. Is that how do you get to those big number of letters across the top?
Yeah. So the way you do that is by each possibility. So let's go back here. So this person can have a big F and a big S, and that's pretty much all that they can give. Because if you have to have an option for your F and an option for your S, and that individual only has big S's and big F's, so that's all they can give. Then you have this individual who has 2 small F's and 2 small S's. So at each case there, we have a small F, small S, and that's what they can give. But because it's a dihybrid cross, we've got more than one option here. So the options we could have, because you can get the big F from the mom or the dad and the small f from the mom or the dad, or you know what I mean, and it can come together. So what you have here is a small f, small s. We talked about that. That's an option. You could get that if the f came from individual on the right and the s also came from the individual on the right. Then you have the option of big F, small s, or excuse me, big S, small f. It's really hard to say with the mask on, I apologize. You have that option here where the big S came from the individual on the left and the small f came from the individual on the right. So all those blue outlines are the different options. So you can either have 2 big letters, 2 small letters, or 1 big, 1 small, 1 big, 1 small. Out of all of these possibilities up here, right here. Right here. Out of all these possibilities. You could get one from one and one from the other. You could get both from one. Those are the possibilities. Then you go through and you look at the crosses of each of those Punnett squares. So if you have 2 big F and 2 big S, you have F, F, S, S, and all the way through. So the more possibilities you have, the bigger the Punnett square. You guys would not have to actually like write this out, just knowing the difference between a dihybrid cross and a monohybrid cross. Did that answer your question? Good. Okay. All right, this is also another way to kind of break it down. It's just to go row by row and then do that 2-trait cross. This is from page 459 and 460. So if you're into patterns, and if you're not into patterns, that's okay, but the patterns can kind of tell a story. So you don't always have to do a Punnett square if you don't want to. But for some people, it's easier to just draw it out and look at it every single time. So with some of these patterns, if you have a monohybrid cross with a monohybrid cross of big A, small a, big A, small a, every single time is going to be a 3-to-1 ratio dominance. That's what that top column or that top row says. If you have a big A, small a, and a small a, small a, it's going to be a 1-to-1 ratio every single time, no matter what letter you're using. So if you're into like understanding some patterns and not doing the Punnett square, this can help you. But if that's taking it 2 steps forward, don't worry about this slide. All you really need to do is be able to maybe write down a Punnett square and then fill in the blanks and tell me the ratio. But if you're like, oh, that's so many steps, I don't want to do that many steps, worry about these patterns in table 21.1. Okay, so now we have different patterns of inheritance. Patterns of inheritance mean how things are passed down. So some things are passed down on your autosomal chromosomes. Your autosomal chromosomes are the ones that are your normal chromosomes, all of them but your sex chromosomes. You've maybe heard of your X and your Y chromosome or your X and your X chromosome determining the sex of an individual. This autosomal word means all of your regular chromosomes, the opposite of sex-linked. So you can have an autosomal chromosome and a sex-linked chromosome. Then we have the type of disorder, so you can have autosomal recessive or autosomal dominant. The steps I like to use for these are to figure out what it looks like to have the trait. So 1, what does it look like to have it? Work our way down. And fill in the blanks. Writing it on the board, on their board. Okay, so what does it look like to have an autosomal dominant trait? Well, if you want to look ahead on the PowerPoint, you can. So we have the option of autosomal. And we have the option of X-linked or sex-linked. And from that, we can have 2 options. We can have dominant and recessive. And same thing with X-linked. You can have dominant and recessive. What does it look like to have an autosomal recessive trait? It looks like small a, small a. You can use any letter here, what works for you, okay? I'm not sure how much of that you guys can see on the board. So let's go to the whiteboard and I'll type it really quick for those on. Okay. So you can have autosomal and you can have X-linked. From that, you can either have dominant or recessive. And same with this one. You can have dominance or recessive. Okay. And then what does it look like to have an autosomal recessive? It is small a, small a, small f, small f, small a, small h. Okay, those are all of those options. So going back to the PowerPoint, for this problem here, this is an autosomal dominant disorder. So what is it? Oh, wait, did I go one too far on this one? There we go, autosomal recessive disorder. So we already determined what it looks like to have it. If you have it, it looks like this. Okay, if you have it, it's small a, small a. Then we have to kind of walk through what this thing is called. It looks like a kind of a bunch of mess if you don't know what you're looking at, but this is called a pedigree. And the pedigree is kind of like a family tree. So if you ever had to do that when you were in school, the horizontal lines, they indicate that the two individuals mated. Now remember, we're only worried about the science here. If somebody got married and divorced 17 times and then only had offspring with one person, on this pedigree, I'd only see that one person. This is not a social tree. This is a genetic and biological tree. So up at the top here, we have a male and a female. Females are indicated by circles. Males are determined by squares. The horizontal line between them means that they mated. Then you go down and branch, and their offspring are the ones that I'm drawing on there. So we have a horizontal line between those top two. That means that they mated. And then here you go down. Those are their offspring. Okay, we don't worry about the social part of it. We worry about the biological. So it doesn't matter if they're married or not. It doesn't matter if they're divorced or not. It just means, did they have some offspring or not? Okay. The females, like I said, are circles. Males are squares. So here, a key, it says that if you have this autosomal recessive disorder, if you have it, you are small a, small a. In this case, we have some people or some individuals who do have it, and they are shaded in as yellow. When we look at autosomal recessive, we do not care about which sex the individual is. Okay? Here's some tips about autosomal recessive disorders. The affected children can have unaffected parents. And we see that here in the middle. We have two unaffected parents, two parents that are not shaded yellow in the middle, but they have some offspring that are yellow. And this is the case, and we've seen that before. When in doubt, draw it out. So draw a Punnett square. This is a big A, small a, big A, small a. Then in the bottom right-hand corner of that Punnett square, they can have individuals that are small a, small a. Heterozygous, big A, small a, have an unaffected phenotype because they do not have small a, small a. Two affected parents will always have affected children. So we see that, this is not an example of that, but that would be if there were two parents that were small a, small a. Oops, that's supposed to be an equal sign. Let's just cross that out. I don't know why I keep trying to draw stuff on there. Okay, so if you have two parents that are small a, the only result that they can have are small a, small a children, sorry for that, that are both female or male. Close relatives who reproduce are more likely to have affected children, and both males and females are affected equally in this pattern of inheritance. Okay, so we're not worried about the sex. Both males and females can have this quite equally. Now we have the opposite of that, autosomal dominant disorder. Our first step is to figure out, what does it look like to have it? If you have an autosomal dominant disorder, you can either be big A, small a, or big A, big A. That is what I'm showing here. So you could either be big A or big A small a, and vice versa for all the other letters. Okay. For this one, we kind of walk top to bottom and we look at it. The females are circles. If they have the trait, whatever we're looking at, because it's autosomal dominant, they just have to have one of the big letters. So it's possible if there were blanks in this and we saw that one was shaded, it's possible that we'd only know that first letter. And it's okay to only know that first letter. That's kind of what it says right here with that A and the question mark, which of course you guys can't see in class because that would be too easy. So that A with the question mark means we know that they have this autosomal dominant trait, but we don't know what that second allele is. And sometimes that's okay, but we just have to maybe walk through some other options. So some fun facts about this autosomal dominant disorder is that the affected children will usually have an affected parent. Okay, at least one of their parents has to be affected by this if it's dominant. It's possible, where do I see that? So on the second generation here in the middle, one of the parents does not have it, and the one parent does have that trait. And even though they have a parent that doesn't have that trait, they can still have the possibility of having it here. So that's an example of that. Heterozygous are affected by this trait because it's dominant. The two affected parents can produce unaffected children. We see that here at the top. The first generation here that we're looking at, these parents have the trait, but then they have a couple of offspring here that do not have the trait. So here's that option right here. They both are affected, and then they have some parents here that, or some offspring here that are not affected. Two unaffected parents will not have affected children. So we see that right here. There's no possible way for anybody in that branch of the tree to have the genotype or the phenotype because there's no dominant allele to pass on. Both males and females are affected equally with this one because we're not worried about gender. Okay, autosomal recessive disorders of interest. So these are just some examples of what that autosomal recessive disorder could be. We have Tay-Sachs disease, maybe you've heard of that. This is a lack of enzyme that breaks down a fatty acid in lysosomes, and it results in accumulation. Another fairly common one is cystic fibrosis. This is when your chloride ions do not pass normally through the cell membrane, resulting in a thick mucus in the lungs and other places, and it often causes infections. So these are different types of autosomal recessive disorders. Autosomal recessive, meaning that they affect males and females equally, and then that whether or not it's autosomal or whether or not it's dominant or recessive is saying what it looks like to have that trait and how it's passed down. Okay. Another example of a couple of autosomal recessive disorders is PKU. This is another lack of enzyme that's needed to make certain amino acids, and this affects the nervous system development. Okay, remember that amino acids are the building blocks of proteins. So if we lack an enzyme needed to make those proteins, we can even just assume that there's going to be some sort of lack of function. Another example of an autosomal recessive disorder is sickle cell anemia or sickle cell disease. This is what we talked about last class period. This is when those red blood cells are sickle shaped rather than that bioconcave sphere frisbee looking shape, and it results in clogged blood vessels. So I'm not an expert on Tay-Sachs disease, but here's a picture of the neuron and how it's affected by Tay-Sachs disease. You can see that there's normal lysosomes in the top picture, and on the bottom we have this bulging lysosome, and it affects the neuron, it affects the function. Think about just like if there's a roadblock on your way to the post office, it's going to affect you getting to send that message or that mail. So here's some examples of autosomal dominant. not sex-linked, but if you have it, you show the dominant trait. You have that dominant allele. Some of those are Huntington's disease. That one's fairly common. This is when Huntington protein has too many glutamine amino acids, leading to progressive degeneration of brain cells. If you have the degeneration of brain cells, it can cause lack of sensation, lack of function, and lack of neurological behaviors. You have osteogenesis imperfecta. This is a defect in collagen synthesis. So making those fibers, those collagen fibers that we find in like your hair, your bones, your nails, your teeth. This results in weakened, brittle bones, which kind of makes sense. Also, the word osteo is a kind of a code word for bone. Then you have Marfan syndrome. This is a defect in the production of the elastic connective tissue, which we talked about, in the protein fibrinogen, and it results in the dislocated lens, long limbs and fingers, and caved chest and weak wall of the aorta. We know from our other lectures and things like that what some of this would mean. So if we had a caved in chest and a weak wall of the aorta, we just talked about that the aorta helps pump blood to the rest of your body. So again, if you're not getting blood to the rest of your body, this probably would make your muscles not be as big and strong. You probably have a lack of mobility and function because you're not getting as much oxygen and as much blood because of that weak wall of the aorta to the rest of your extremities. Okay, here's a slide about Marfan syndrome. It says the connective tissue defects. So these are just some of those defects that I was talking about. You can have skeletal defects. You can have defects in your heart and your blood vessels, which is exactly what I was talking about a little bit there. Your eyes and your lungs and your skin all can be affected because of Marfan syndrome, which is an autosomal dominant disorder. If you have some disorder because of this, your skeletal system can be affected because of the long, thin fingers and arms. You could probably have some flat feet issues, long and narrow face, loose joints. This is structural things that are wrong with or not normal in that body. Then we talk about the heart and the blood vessels. This is a little bit talking about that aorta, and that if you have a weakened wall of the aorta, it will start to enlarge. And because of that, you can have an aneurysm or an aortic wall tear. Just think of that wall or that aortic valve in your heart and it just being weakened. It's going to cause some malfunctions there. Your eyes, your lens dislocation, you can have severe nearsightedness. Does anybody know what the difference between nearsighted and farsighted means? Anybody a contact or glasses wearer? I wear contacts. I am nearsighted, which means that I can see near. That's how I remember that, which means that if I were to take my contacts out, I couldn't read that poster. I probably couldn't read your shirt. I'm pretty severely nearsighted, but I do not have Marfan syndrome. This can also affect your lungs, having collapsed lungs. A collapsed lung is thinking of a lung that does not expand and contract with the breathing. And then your skin can have stretch marks or recurrent hernias, a dural elastica, stretching of the membrane that holds the spinal fluid. So Marfan syndrome is a syndrome, and it can, even though it has one trait, it can affect multiple things in that person's body. So different genetic disorders. Here's an example of Huntington's disease and cystic fibrosis. Cystic fibrosis is that when the mucus gets caught in the lungs and other parts of the body, causing infection, trouble breathing. A percussion vest is something that somebody can wear when they have cystic fibrosis, and it kind of, they say percussion, it kind of is like a drum or like a percussion or a beat or a vibration in that vest that's going to kind of break up that mucus in the chest and in the lung area that allows for that person to breathe a little bit better. Huntington's disease, you can see the top, that is a picture of some neurons in the normal brain, and that bottom picture is a loss of neurons because of that Huntington brain. That middle picture is just talking a little bit more about that cystic fibrosis. And when you have a defective channel, you're not allowed to move the chloride ions in and out of that cell as well as we'd like to, and it ends up causing a thick mucus. If we were allowed to, and if you don't have cystic fibrosis, your chloride ions are going into and out of that cell fairly easily. I kind of think of like what it would feel like to have, when you have cotton mouth, when you've been outside or you're working out a lot and you're kind of dry, feel like that mucus is forming. That's kind of what that is like for cystic fibrosis, but on a different level. I'm not trying to belittle that at all. Pre-implantation genetic diagnosis. So this is a little bit talking about like genetic counseling in a sense. So if parents are worried or individuals are worried about passing on an allele for a genetic disorder, they may do genetic counseling in that sense. Or there's this thing called in vitro fertilization, which is also called IVF. And this is where they can kind of go in and change a few things of the genetics because they can pick and choose. Now there's some ethics to this, right? This isn't saying that this is the right thing, and this isn't saying that it's the wrong thing to do. But this is just something that is possible. They seek assurance that their offspring will be free of this disorder. So maybe somebody in their family has cystic fibrosis, or maybe one of their previous children has cystic fibrosis, and they would like to ensure that their second offspring does not have cystic fibrosis. Okay, that would be something that would go into this. It says only embryos that will not have the genetics or disorders of interest are placed in the uterus to continue developing. So here's a picture of that. And like I said, this is just discussing the science. It's not saying whether or not this is the correct or ethical thing to do for both sides. This is an 8-cell embryo. You get one of the cells that is genetically healthy, and you use it to fertilize the sperm and egg cell, and then the egg is genetically healthy, and then it is injected into the uterus where it continues to grow, and then it says the embryo develops normally in the uterus. And also, these 8-cell embryos can also develop normally in the uterus. The word polygenetic inheritance, you can look at that root word and see the word poly. Poly meaning many. The opposite of poly would be like mono or di. Polygenetic inheritance is 2 or more sets of alleles that govern one trait. So obviously there's a spectrum of different skin colors and tones, and then there's that can range from varying to dark to light skin. That also is the same with height. There are varying amounts of different heights of individuals, of full-grown adults, right? This is because of a polygenetic inheritance. It doesn't mean that when you have big X, small x, that you are going to be 5'7". No, that doesn't mean that. It just means that at that trait, there could be a spectrum of things that are involved there. It can be a couple of different things. Each dominant allele codes for a product, so these effects are additive, and the results in a continuous variation of these phenotypes. That's why nobody really has the exact same skin color. It's a spectrum. The environmental effects can also cause intervening in phenotypes. This is that nature versus nurture a little bit here. The nature behind of it says we've got a certain amount of traits and that's what we can pass down. The nature of it here is you have certain genetics. If you think of an individual who was malnourished, who didn't get enough nutrients, compared to somebody who got ample amount of nutrients, and they both have the same genetics, the 2 individuals could still be different because of their environment that they're in. It doesn't just mean that you have big X, small x, and you're going to be 5'7". If you weren't nurtured or didn't have the right nutrients or things like that, Maybe that person didn't grow and develop in the way that they should have or could have. The possibility, should and could is a different word. Okay, so this is polygenetic inheritance. Another way to look at that is a multifactorial trait. So skin color is also influenced by sun exposure. Height can be affected by nutrition. So that's what I was just talking about. So whether or not you're getting a nutritious food can affect your height. That's why most parents are kind of sticklers about what their kids are eating, right? They want them to succeed at whatever that means or be as healthy as possible, because nutrition and the things that you do to your body can affect those things that are polygenetic. Skin color is also influenced by sun exposure. This is why in different types of the world, you'll see as you get closer and closer to the equator and where there's more sun exposure, the individuals have darker skin colors than somebody maybe who is native to where there's less sun exposure. It doesn't mean though that if I go live in Ecuador that I'm just going to, my skin color is going to genetically change. That doesn't mean that. It just means over time and evolutionarily, skin color has been affected by sun exposure. Okay, here's another way to look at it. It's kind of that spectrum. It's like a graph. Okay, you can have the different types of skin colors, and even on this cartooning graphic, it still is trying to put skin color and things like that into a graph, and even that's not true. Like we can't match up our color to that, but it's kind of showing you that there's some kind of less common and more common, that bell curve, and this is just percentage wise and not anything other than numbers. And then it says that, like, for example, the one with height in the middle, the height in inches, it's kind of rare to have somebody who's like extremely short, and it's rare to have somebody who's extremely tall, right? And then there's kind of like that middle ground, okay? And this is just showing that in inches. Same thing with kind of the color of someone's skin. It's really rare to have albinoism, okay? And then on the opposite end of that, it's very rare to have extremely dark skin, but then there's like a spectrum in between, right? And it's not like you can define that. And it's just showing that these are polygenetic inheritance. Okay, demonstrating environmental influences on phenotype. So this is how we can show, oops, sorry for that, that the environment influences that phenotype. This is a Himalayan rabbit, and their coat, the color of their fur, is influenced by temperature. There is an allele that is responsible for the melanin produced that appears to be active at lower temperatures. The extremities have a lower temperature, and thus the ears, nose, paws, and tail are in darker color. So this is where the environment is affecting what the trait of that rabbit looks like. This is just an example. This is figure 2115 in your book on page 21.15. Then there's something called incomplete dominance. This is another term. This occurs when the heterozygote phenotype is an intermediate between the phenotypes of two homozygotes. So let's say that curly hair trait is indicated by two big C's, okay? And maybe straight hair is considered two big S's. And then if you have a heterozygote phenotype, this is saying you kind of got some wavy curly hair, but you kind of have some straight hair too. This is that intermediate between it. It means that the dominant traits are still being shown, but they're just incomplete. They're just kind of there. So the example here would be like wavy, not exactly like curly Q, but not straight hair. There's a kind of like a middle ground, that intermediate, incomplete dominance. And we don't need to know that slide. Then there's something called codominance. And this is an example of like blood type. That's why our blood type A and B are both dominant, but they are co-dominant together and they exist together equally. So you could have a parent who is type A blood and their alleles are AA, and you could have a parent who is type B blood who is capital B, capital B. They have offspring. It's possible that they could have offspring that are AB blood type. This is called codominance, when the alleles are equally expressed as heterozygote, meaning that they're different but equally expressed. The opposite of this would be that, like with a dominant and recessive trait, if you have something that is big D, little d, that is not codominance. This is codominance because they're both expressed equally. All right, multiple allele inheritance. This is just saying that there's a gene that exists in several allelic forms. A person has two of the possible alleles. And again, this is an example of ABO blood typing. So the A and B are co-dominant alleles, but then there's an O allele that is possible. And it's recessive to both A and B. So this is considered a multi-allele inheritance because we have codominance with A and B, and then you have dominant and recessive with the A's and B's, and then compared to the O's. So for this, if you have O blood type, you must have two O's in that location. But if you have type A or type B, you could be AB, you could be BO, AO. When you have B and O together, you're type B, but your second allele is an O. When you're big AO, you're type A, even though you have that O allele. And then if you have two O's, you're type O. This comes into play when we start to think about inheritance of the blood type. You could have two parents who are type A, and then one is type B, and they have an offspring who is O. And this is possible because they could be AO and BO. The other way to look at this is they draw it with eyes. You don't have to draw it with those eyes. You see the letter I? You could just write an A here, and that makes sense to me. And hopefully that makes sense to you. You can also just draw an O here. And I'll draw that here on this board. So you can also write it as AO and BO. And then looking at those crosses, it's possible that you could have an individual that's AB blood type. You could have an individual that is B. You could have an individual that is A. And then you can have an individual that is type O. The phenotypic ratio would be 1 to 1 to 1 to 1. In this case, you have one that would be type AB. You have one that would be type A. You have one that is type B, and you have one that is type OO. Okay, rewind our brains a little bit. We talked about autosomal patterns of inheritance. The opposite of autosomal is your sex-linked inheritance. These are your X and Y or your XX chromosomes. These are the traits that sex matters, the type of individual. So maybe you've heard of like color blindness being more common in males, or like male pattern baldness. Those are things that are more common or passed down to because of the sex-linked chromosomes. So when somebody is XY, that is meaning that at their chromosomes, they have an X chromosome and a Y chromosome, and that indicates male as the sex. Then you have an X chromosome and an X chromosome, and this means that it is a female. And this is talking about just chromosomes, not identification. Just wanted to put that out there. This is just talking about genetics here. So we can have something that is passed down on the X chromosome, meaning that if it's X-linked inherited, you can show it on the X, which means if you're a female, you can have it on both X. If it's something as Y-linked inherited, you can have something on the Y, but that means females don't have the option of having that trait at all. This is called sex-linked traits. I call them X-linked, You could also call them sex-linked. Same thing. Okay. So let's talk about this one. It's color blindness. So if you have color blindness, you have to have the recessive allele, okay? I'm going to go back to my table here on the Zoom, but we worry about the sex here. Okay, we are worried about whether or not it's a male or female. So if you have color blindness, which you guys can't really see here, but I could zoom in. Okay, you have to have X, B, Y, and that's a small b, or if you're female, you have to have X, little b, X, little b. Okay, that's what it would look like to have a sex-linked recessive disorder. Sex matters here, so we have to be worried about the gender. Here, remember, the gender wasn't affected. It affects males and females equally. Okay, so let's go back to this big example. We've got parents who are both normal vision. Neither of them have color blindness. You can see that because here we have X big B Y, X big B little, and then X big B, X little b. Okay, I'm going to go back to this whiteboard really quick and update this. I think if I go, it won't let me do it. So on the board, Zoom people, I made those little B's like short and on the corner, but it won't let me do the command for it on Zoom. Okay. So this is color blindness. The parents have normal vision. We can see that because of the shading, but also because of their genotype. You cross the parents. Now here, we still are worried about the X and the Y chromosomes. So here you have X and X across the top here, and you have Y, X and Y here. This is an X-linked recessive disorder, so there's no trait on the Y. You guys can kind of see that here in class. Then you go to each intersection. So the parents were big B and then big B, small b. So here we have a X big B, X big B. So that's what you write in that top left corner. X big B, X big B. The next intersection is X big B, X little b. So far, no color blindness, but a carrier in that sense. Here we have a male who does not have color blindness. They have normal vision here. And then you go to the next intersection, you have X, little b, and Y. And that indicates that this individual would have color blindness. Now there's a way to say that, even though I'm drawing like all over this because it's so hard to draw. There's another way to say this question. If I said, what percentage of the children or the offspring would be colorblind, your answer would be 1/4. Or a 3-to-1 ratio, 25%. If I said what percentage of males are, what's the probability of a male offspring getting this color blindness, you would say 50%, the 1-to-1 ratio. So being a little concerned here about the sex, the gender, right? If I said what percentage of females are color blindness for these offspring, what are the possibilities? It's a 0%. What's the possibility for males? It's a 50% chance. Does anybody have any questions on how I got that number? Or anything about that too? That's open for that. Okay. So this is still talking about color blindness here, X-linked disorder. Color blindness is a recessive X-linked disorder. You can also see in that key there what affected and unaffected look like. Remember, females are circles, males are squares here. We are worried about gender, So in every circle, there should be two X's. In every square, there should be an X and a Y. Whether or not they're shaded or not shows that they are affected by color blindness. You can also see in this case that there is a female who's affected by color blindness. What does that mean about her mother, or the female? Sorry, I'm going to go to the next slide here. What does that say about this individual's mother? I'll give you some options. Was the mother like this? Did the mom look like that? Did that individual's mom look like this? Thumbs up or thumbs down, Zoom? Does the mom look like that? Nope. The next thing, so we can say that the mom is for sure not homozygous dominant. The mom at least has to be a carrier of that. We don't know if that means that she was dominant or recessive, but what we can see here is that the mother was a carrier. What does that mean of the father, or the male figure here? It means that the father has to be colorblind, which is true. If you look back, the father is colorblind. So this individual here, compared to here, the father has to be colorblind. A female gets one X chromosome from their mother and one X chromosome from their father. A male always gets their X chromosome from their mother, and they get their Y chromosome from their father. And of course, the words mother and father are subjective. Here's a couple examples of some X-linked disorders. The one that we're missing up here is dominant. If you are dominant, you look like that, or like this. This could be a big B or a small b. Filling that in up here. That blank here for the Zoom, the blank means that it could be big B or little b. Okay. Got a couple more slides, so sticking with me here. These are just a couple of examples of those X-linked disorders. So disorders that are passed down because of an X-linked recessive or dominant. So you have color blindness, you have a certain type of muscular dystrophy, Fragile X syndrome, and hemophilia. So here's talking about that the Dunt muscular dystrophy. Again, I'm not an expert on this disorder, but I can tell you a little bit about the different muscle tissues here. So muscular dystrophy is going to affect the makeup of the muscle tissue, whether that be skeletal, smooth, cardiac, things like that. If somebody has normal muscle tissue, there's that example on the right. So that's that normal tissue. The abnormal muscle tissue on the left, as you can see, is a little bit weaker. It has some fibrous tissue in the middle, so this will affect the structural and the functional parts of that muscle. And lastly, this is just a big family tree here. Does anybody know, without reading the caption, where these names come from? Does anybody recognize any of those names up there? Besides the fact that my name's up there. Anybody recognize any of those? This is the royal families, and this is their family tree because there is a hemophilia disorder that has been passed down through the royal family. You will also notice some great relative mating here in the middle. Yay. Or where is that? Not there. Yes, here. So these people right here, how are they related? Can anybody tell me that? Or do you want me to walk you through it?
You zoom in a little bit.
Yeah, it's kind of hard to see. This is also in your book. So that won the first circle because I'm really bad at drawing on this thing. So it looks like cousins, right? Yeah, they're cousins. So here is Victoria and Edward. Sorry, I don't mean to keep doing that. So here we have Victoria and Edward at the top. Stop doing that. Okay. They have a child named Victoria. Victoria and Albert. They have, as you can see, a lot of kids. They have, how many kids do they have? 1, 2, 3, 4, 5, 6, 7, 8, 9 kids. You guys can't see them all because we're zoomed in here. All of those kids, we have Alice and Louie. Louie and Alice have 1, 2, 3, 4, 5, 6, 7 kids. And then these kids, this is the circles uncle right here. I should draw with my pen because you guys can't see that. So the circle here, and this is their uncle. So this offspring would be their cousin. And we're not talking like second cousin twice removed. We're talking like this is their actual cousin. So that's interesting. You can see that from a pedigree. I don't think that happens again. No. So that's interesting. And when you have incest in the sense of this, there ends up being usually some sort of genetic disorders because of that lack of variation. So this is just a pedigree, a family tree of the royal family, and then it's talking about the hemophilia that is passed down. We talked about in the previous slide that hemophilia is an X-linked disorder. Does anybody have any questions over anything that we talked about today?